Hello everyone, my name is Gautam Chauhan and I'm a research fellow at Pine Biotech, and today I'll be discussing with you about my research topic, that is comparative transcriptomic analysis to understand the role of melanin in melanoma. Before jumping directly into the topic, let us first understand about melanin and melanoma. Melanin, it is a term that we all have heard before. So melanin, as we all know, it is a pigment that is found in most of the organism, and it determines our skin color, our hair color, and even our eye color. So melanin is basically a pigment, naturally occurring pigment that is present in most of the organisms, and it determines the color of our skin, our hair, and even our eyes. Um, it is produced through a process called melanogenesis. Melanogenesis is basically made up of two words. First is melanin and second is genesis, that is formation. So the production of melanin is called melanogenesis, and melanin is produced in a group of specialized cells that are called melanocytes. The main function of melanin is to protect us against the UV radiations, and we can say that if we are more exposed to the UV radiations, there would be more melanin production in our body, and therefore our skin would be of darker color. Basically, if somebody has a darker skin or a darker color complex, then his or her skin would be having more melanin, and that is actually a very good thing, because we'll be learning further in the further slides that having more melanin or having a darker skin is actually beneficial, because it reduces the chances of you developing a melanoma. Thus, um, more melanin production or darker skin, the better it is to protect yourself from melanoma. So next, if I talk about melanoma, so melanoma is one of the types of the skin cancer, and it is the most serious type of the skin cancer that develops in the melanocytes. Melanocytes, we discussed earlier, that the granules in which the melanin is produced. Um, so it is the most serious type of skin cancer. There are other types of skin cancer as well, but melanoma is the most serious type of skin cancer. And as you can see in this picture, it will look something similar to this. Initially, you might think of it as a mole, but when it starts spreading, uh, it obviously would not look like a mole anymore. So melanoma is basically the most serious kind of skin cancer, and one of the risk factors that I told you is having a fairer skin. Yes, if you have a fairer skin, the chances of you developing melanoma is very high. So it is one of those risk factors. Risk factor, we all know risk factor basically means anything that would increase your chance of having that particular disease. Thus, having a fairer skin would increase your chance of developing melanoma. So now that we've learned about melanin and melanoma, let us talk about our topic, that is about the transcriptomic analysis of the role of the melanin in melanoma. So, if I talk about the human epidermal melanocytes, or the HEMs, they provide a critical protection against the skin cancer. Again, skin cancer are of different types, but here we'll talk about melanoma. It protects us against the sunburns and photoaging. So they definitely play a very important role in skin cancer or melanoma. So in this um, research project, I took two samples from the darker skin people and two from the lighter skin people, and then compared them to understand about the different genes that are expressed in the darker skin and lighter skin. And as I discussed earlier, that the darker skin people would be having more melanin production in them, thus their chance of developing skin cancer is lesser, and the lighter skin would be having very less melanin production in them, thus their chance of developing skin cancer or melanoma would be higher. So this was the basis of my study. This was the basis on which I started about my research project. Now, if I talk about the methodology, from the data set that I got, I initially ran the RNA sequencing pipeline to get the expected gene count table. This was my result that I got. A lot of results, but the important one was the gene uh, expected count table. After which I moved on to the PCA, and finally I developed a pipeline for the differential gene expression, and through which I got very good results, which I'll show you in the later slides. And that helped me to interpret the level of expression of the genes and how those genes were expressing in the darker versus the lighter skinned people. So this was the first plot that I got after my differential gene expression pipeline. In this is basically a volcano plot that shows us the levels of the gene expression. The red color genes that you see on your screen is the genes that are having higher level of expression in them. On the x-axis we have log to fold change, and on the y-axis we have the p-adjusted value. So we have a variety of genes. The red ones are uh, more differentially expressed than the black ones, and the point that we see on the x-axis, if it's on the left or on the negative side of the x-axis, it would be down regulated gene, and on the positive side or the right side of the x-axis they would be upregulated genes. Another graph that we got from this was about the mean of the normalized counts against the log fold change. In this also we see the blue dots are the ones that show high level of expression. The gray one were the normal level of expression, we can say, and the blue ones were definitely higher. Also in this, all the values above 0, like the positive values, will be for the upregulated gene, and the negative values would be definitely for the downregulated genes. This was another important result that we can interpret from this graph. Next we had the gene ontology enriched pathways, in which we can see different pathways that are present against the gene ratio on the x-axis. So we see the different pathways like the cell junction assembly, the sodium ion transport, skin development, positive regulation of the cell junction assembly. So these are the top 20 pathways that were present, and the red dot signifies that it's having a higher level of expression, and the dot value basically shows, or the circle would show that how many number of genes are present. For example, if I talk about the cell junction assembly, we can see it would be having around 8 genes, and its p-adjusted value is nearly about 0.03.02, and it is highly differentially expressed. So this was the type of interpretation that we would be getting from these graphs. And next, what I would be doing is basically taking the genes that are quite highly expressed in the melanoma cells, and then I'll be comparing it with the fairer and darker skin to see whether those genes that are involved in the melanoma or the melanoma pathway are highly expressed, or highly present in the darker skin or lighter skin. And then I'll be concluding with my work stating that these genes have this much level of expression in the darker skin and this much in the lighter skin. Obviously those genes would be having, if a person has melanoma, then definitely those genes would be the genes that would be present in a less quantity in the darker skin, and probably in more quantity in the lighter skin. But when I'll do that analysis, that would be the end of my work, and then I'll be concluding my work. Thank you. I hope you understood this presentation and the work I did till now, and I'll be continuing it further. Thank you.